thank you for the introduction and for the opportunity to speak here. I am going to be talking about some of our work on spindle-nuclear envelope coupling and closed mitosis. But before we dive in, I just wanted to mention something that was inspired by one of my favorite moments in the meeting so far. Yesterday, when Wenjing told us we could have used machine learning, but we used human learning, okay? I really like that. And this talk is going to be very much in that spirit, okay? We mostly have a modeling focus. But this comment reminded me of one of my favorite commentaries on work in this area, okay? So this is an XKCD comic. And the tip is, when you do a task by hand, you can say that you trained a neural net to do it, right? Okay, so Wenjing, you could point that out. So all of the work in this talk involves trained neural nets, okay? Technically, right? So that's what we're doing. Okay. So this is the group, and I'm going to be talking about work from people in my group. I'll mention as we go along. But a bunch of this talk is a collaboration with Mary Elting and with Sharonda LeBlanc, who are at North Carolina State. And so I'll mention their work as we go along as well. And this work also involves quite a bit of work that is in progress or preliminary. Most of this I haven't talked about before. So you'll hear some pieces that we understand more and some pieces that we understand less, and hopefully that makes things interesting for discussion. But before I talk about the spindle, I was just going to mention a couple things we're working on or doing recently that I'm not going to talk about that are maybe of interest to people here. So the first is a recent paper that we had that was doing some modeling of some really beautiful work from Scott Forth's lab, where they looked at microtubule pairs that are cross-linked by cross-linker PRC1 and then slid apart by motors, kinesin motors on a surface. And they had in this nice work from 2021 a result that this can happen in what looks like two different states. So sometimes the resistance from the cross-linkers makes the filament slow down a lot, and sometimes it makes them slow down not as much, right? There's higher and lower resistance. And in recent modeling work, we found that we think that that's controlled by different geometric states of the microtubules, that they can be in a further separated state where the cross-linkers aren't tilted a lot, like in the left here, or they can be in a closer together state where the cross-linkers are tilted more, and there's a distinct transition between those states that controls the friction. And this has gotten us kind of interested more generally in how lateral separation between microtubules affects their organization. And related to that, we've been fascinated for quite some time about how this observation from Dick McIntosh in the 90s, that in the yeast spindle, you can get a square, if you look at the cross-section, you can get a square array of microtubules that are oppositely oriented. So it looks like an antiferromagnetic crystal. And that's very striking. And in addition, if you look at the typical separations between these microtubules, there are 15 for antiparallel and 32 for parallel microtubule pairs. And that's a surprisingly small distance relative to the size, as best we know it, of the proteins that do the cross-linking and sliding and organization of those microtubules. And we have some work in progress where we think we can understand both what sets this separation and why it's those values, and also how you can have a model that makes this nice antiparallel square array emerge. Okay, but that's to talk about later if we have time in our last couple hours of the meeting. All right, so let's talk about the nuclear envelope and mitosis. And so if you've learned about mitosis in animal cells, most of the time we see pictures that look like this, okay? So what everybody knows is that the chromosomes are inside the nucleus during interphase, and then at the onset of mitosis, there's nuclear envelope breakdown, and the spindle assembles in the absence of constraints from the nuclear envelope, okay? But that's open mitosis, but that doesn't happen in every organism, okay? And we're going to talk about closed mitosis, and we're going to talk about the best yeast. Julian, I'm really glad that you said that. We agree with you. We also like the best yeast, so fission yeast, because Schizosaccharomyces pombe. And here's kind of the cartoon version of what mitosis looks like in this organism, where blue shows the nucleus that remains intact as the spindle assembles and segregates chromosomes. And on the right are some imaging data showing what this looks like in the microscope. So I'll show a lot of images like this. Typically red will be labeling microtubules. So this red line is the spindle over time. And here in these images, the green is a protein that marks the nuclear envelope and also spindle pole bodies, the ends of the spindle in early mitosis. And so you can see that during chromosome segregation, the nucleus undergoes this beautiful set of shape transitions where it first elongates, then forms kind of a peanut-type shape, then sort of a dumbbell that looks like two sort of roughly spherical things separated by a bridge, that eventually the bridge breaks down at the end of mitosis. And so I think this is a beautiful problem where you have to look at coordination of mitosis with other things that are happening in the cell. So in this case, chromosome segregation and the separation and ultimately division of the nucleus and the nuclear envelope have to be coordinated together. Okay, and that's what we want to try to understand. All right, so given that this happens in the best yeast, of course we want to study it because it's the best. But sometimes, hopefully not from a lot of you, but sometimes people will say things like, well, what I really care about is humans, and humans don't do this, and I want to cure cancer, and that's all open mitosis, so I don't care, go away, I don't want to talk to you, right? So I want to try to, hopefully don't think that way, but if you do, why, even if you're going to cure cancer, you might care about this, okay? And so one is that if you are interested in the spindle, like the pictures I showed in the beginning, when we talk about the mitotic spindle, we very often have cartoons like this, where there's the cell, there's the spindle, there's the chromosomes, there's nothing else there. It's just sort of floating, no other components of the cell, right? But we know that that's not the case. Vijay talked about this a little bit. People who talk about spindle positioning and spindle orientation get at this somewhat. That spindle does interact with the rest of the cell, and having model problems for how that works and how we coordinate maybe can be a nice thing to have. I'm also going to try to make the case that the interactions between the spindle and the nuclear envelope are going to let us learn things about the mechanics of both of them that aren't always easy to get at for the spindle. And you might be interested in also thinking about mitosis comparatively and thinking about how do cells adapt when they do mitosis in different ways, and that might be interesting. Okay, you could also potentially be interested in the nucleus. And compared to the cytoskeleton, the mechanics of the nucleus and of the nuclear envelope are much less well understood. And I think that's quite interesting because there are a lot of open questions about even what are the right physical descriptions that we should use. And we here have a nice system where the spindle can really make big mechanical perturbations to the nucleus, and that could help us understand that. And then it turns out that even if you don't think closed mitosis is that interesting, the principles of how the nuclear envelope gets remodeled are quite conserved across the best yeast and animals. They just happen at different times, and we should still be able to learn interesting things. And then finally, I think an interesting direction for our field to go is to think about integrative modeling that can build in more components of the cell that we might want to understand, right? So in this problem, we have to think about the cytoskeleton. We also have to think about membrane biophysics. And I'll argue that polymers are going to be somewhat important in thinking about chromatin as well. And this maybe can help us ask new questions and think about new things. Okay, so that's my pitch for it. Okay, so the first part, I'm going to give you a little bit of orientation to prior work and what we already know about closed mitosis and spindle nuclear envelope interactions. And here's some kind of orientation to the fission yeast mitotic spindle. So I already showed you some images of what it looks like. Here's some nice work from about 20 years ago doing electron microscopy reconstruction of the spindle. And I have to say, I feel a little bit embarrassed putting this up after Stephanie's really nice talk where there's tens of thousands of microtubules and all this amazing work, okay? We have about 30 microtubules, all right? Max, okay? It can be fewer than that later in mitosis. There are 3 chromosomes, but that can have some advantages that I'll try to talk about. It's possible to, even way back in the 90s, do EM on them and get an idea of the structure. The gray here is showing the nuclear envelope, okay? And the yellow, spindle pole bodies, so SPBs, that's the yeast term for centrosomes. The SPBs are embedded in the nuclear envelope, and all of the spindle microtubules are nucleated with their minus ends at the SPBs. So we know a lot about the organization of the spindle and where the microtubules come from, okay? We have this strong coupling to the nuclear envelope. Fission yeast also does not have lamins, so that can help simplify our understanding of the mechanics and how we think about it. And so this kind of relative simplicity, I think, makes this spindle and the system appealing for modeling of the type that we do, where we want to kind of put in all the key particles and their dynamics and try to work out what we can predict. Okay, and so this, in fact, is something that we have been doing over the years for more than about 10 years now, I got very interested in thinking about, in this system, how can we understand the self-organization of the bipolar spindle? So related to what Jing just talked about, but thinking from a more, okay, we know there are certain motor proteins and cross-linkers, there's microtubule dynamics, there's SPBs. What are the minimal rules you need to get a bipolar spindle? So we did work on that. And then we extended a step further to say, how do you get chromosome attachment to the spindle and error correction and alignment at the same time that this bipolar structure is self-organizing, and getting proper bioorientation and then ultimately segregation. And Robert and Chris worked on that in the lab. And so this was great. We really were happy about this work, but it always kind of bothered me that the gray line here is showing a fixed spherical nuclear envelope. And we didn't keep it completely rigid. We made sort of some approximations to what would you estimate would be a force from the nuclear envelope if things move a little bit radially, sort of like a radial potential. But it wasn't very satisfying. And a model like this certainly is not capable of capturing the dramatic shape changes that I showed you before. And in the context of doing that, we also found that, that this force that's applied from the nuclear envelope on the spindle in our model was quite significant in helping set what the length of the spindle was. And so I was really interested in eventually kind of like going beyond this and having a better model that includes the nuclear envelope in it's kind of a real way. Okay, so that's sort of one motivation. There's some other really interesting observations that helped inspire this work from the Olafarenko lab and this paper, which I think is really quite beautiful from Current Biology. They quantified that over the course of mitosis in pombe, in our fission yeast, the area of the nuclear envelope increases. Okay, the nucleus keeps approximately the same volume, but its area goes up about 30%. And this is really important. It's required for mitosis to work properly. And part of the way they could look at this is they found that this area expansion depends on fatty acid synthesis. So the cell is actually making new lipid molecules and using that to make a bigger nuclear envelope. And one way you can see that very dramatically is mess it up, okay? And so in these experiments from their work, look especially at this lower panel, they can treat the cells with a drug called cerulenin that's an inhibitor of fatty acid synthesis. So when the cells are treated with this, the nuclear envelope area cannot expand during mitosis, okay? And look at what happens. I think this is so amazing. So here, red is marking the nuclear envelope and green is marking the spindle. The spindle elongates, and you see it can't deform the nuclear envelope into this peanut and dumbbell like we would usually see. And instead, the spindle actually buckles, look right here, at about 8 minutes. It's bowing, and then it breaks in half. Okay, the spindle actually breaks and then comes back together into a single bundle. So this, I think, is very, to my mind, compelling evidence that, that mechanics from the nuclear envelope, especially when it's messed up, can have a significant mechanical effect on the spindle. Yeah, question?
Kind of membrane surrounding because they look like
yes.
they're protruding out.
Yeah. So down here, where it looks like there's just green sticking up, the nuclear envelope is very, they don't show it directly here, but it's very likely distorted around the spindle. And I'll show you why later. So hold that thought, but I do not think that it's rupturing. Okay, yeah, good. Okay, so I thought that was kind of really remarkable and I wanted to understand it. So let me just give a couple highlights. Okay, this is what normally happens. What other things can go wrong if spindle and nuclear envelope aren't coupled correctly? Okay, so I just showed you this one. If you reduce the area expansion, force from the nuclear envelope, we think, can cause the spindle to bundle and break, buckle and break, and that can cause a failure of chromosome segregation. It's possible to go the other direction, okay? You can perturb fatty acid synthesis pathways in a direction that leads to overproliferation, too many lipids. The nucleus gets larger and it looks floppy, and then the nucleus gets misshapen and it looks like, this is preliminary, but that you can get sort of an over-elongated spindle in that case. It's possible to perturb kinetochore attachment to the spindle. The kinetochore people in the audience will know a lot about this. If you do that in fission yeast and chromosomes are unattached, then when the spindle elongates, it doesn't form the proper peanut and dumbbell. Instead, it just makes tethers, like thin kind of membrane tubes at the sides of the nucleus. And then in a really remarkable experiment with a couple of genetic perturbations, This is unusual, but it's possible in yeast to make an acentrosomal spindle. So the spindle assembles, but it doesn't have the SPBs, so they aren't embedded in the nuclear envelope the way they're supposed to. And then again, when the spindle elongates, it makes these nuclear envelope tethers instead of the dumbbell. And then a really amazing thing happens where the spindle elongates and anaphase A happens, so the chromosomes are attached and they attempt to segregate, but they aren't able to get to the ends of the spindle. And the hypothesis put forward in this paper is that they just can't fit into these thin nuclear envelope tethers, right? So the morphology not changing the correct way in the beginning then disrupts chromosome segregation, even though the processes are happening kind of the way we think they're supposed to. Yeah.
So in the acentrosomal spindle, is there any other mechanism that focuses the microtubules to where the SPB should be?
Oh yeah. So what kind of clusters the spindle, the microtubules? Yeah, there are. We should talk about that afterwards. Yeah. There are cross-linkers and motors that help organize it. Yeah. Okay. So this result in particular, I think, is really remarkable for how like disruptions of this coupling and the morphology then can kind of cause problems for you. So all of these themes seem like interesting motivations to study this. And there has been some work trying to look at the nuclear envelope morphology. And in particular, some nice work from Shelley Sazer and Greg Huber quite some time ago, almost 20 years ago at this point, showed that a vesicle-like model of the type that we heard about from Julian, characterized by a Helfrich energy where the nuclear envelope has resistance to bending and it has a surface tension, and you can also include kind of volume constraint terms, can characterize the shape fairly well. Okay, so they did calculations of what would the shape look like with a nuclear envelope with sort of a range of estimated parameters and kind of a constraint of a microtubule bundle on the inside of it, okay? And they did energy minimization, assuming an axisymmetric shape. And one of the things they found that got me interested in this problem is that they can get very nice tethers, right? Those long tubes, but no dumbbell comes out of this, right? And then they even went a step further and compared their model to some electron microscopy that characterized the shape. So this dashed line is showing the shape of the nuclear envelope in an EM section with the presence of one of these microtubule bundles. And they looked through their parameter sweep which shape gave the closest match to what was observed experimentally. And what they were able to constrain is not everything, but it's still something. Really, the ratio of the surface tension to the bending modulus is what they could select. And they get a particular value for that. And then they did some additional estimates like, okay, if you say the bending modulus is about 40 piconewton nanometers, which is about 10 kT, that's a kind of reasonable range for what's seen for bilayer vesicles, then the tension would have this value of about 100th piconewton per nanometers. Again, what really the bending modulus of the nuclear envelope should be is not that clear. It's not a bilayer membrane, it's a double bilayer, and it has this other structure, but at least, we're kind of working towards trying to do something quantitative here. And so I've kind of worked based on these values because at least it's a starting point. But again, no dumbbell, right? So then there were a couple of really nice papers that came more recently that said, well, given that this peanut and dumbbell shape doesn't happen if you don't segregate chromosomes, a pretty reasonable hypothesis is that having the chromatin segregated to near the poles might distribute force from spindle elongation over a bigger area. And maybe that's what controls the shape. Okay, and so in this paper from Zhu et al. In the Biophysical Journal, they varied this parameter sigma, which is kind of what fraction of the area near the pole do you distribute the force from the spindle over. And when that fraction gets big enough, you start to see shapes more like what you see in cells. And then Feng et al. Made kind of an extended dynamic model that also included a representation of spindle elongation and could calculate kind of the shape transitions, again, with force distributed over a bigger surface area. Okay, so that's kind of some nice work that we want to build on. But what would we like to do? What are kind of our questions and our interest in this project? Well, we'd really like to understand this coupling between the spindle and the envelope. So we would like to do things. So for example, like the cerulenin treatment that I showed you from the Olafrenko lab that perturbs the nuclear envelope and see what that does to the spindle and see, can we predict it? And we'd also like to go the other way, right? If we mechanically perturb the spindle, what will that do to the nuclear envelope? And I'd really like to find out what the forces are. Though apparently all this time I should have been talking to Julian. He has really nice ways to do this. I'm starting to regret spending all my time hiding in a cave in the mountains in Colorado and not knowing about what's going on. But okay, still we can try to do what we can do. And I'd really like to know what nuclear envelope mechanical properties are consistent with all this to just better understand physically what the nuclear envelope is like and be able to try to build predictive models of that. Okay, so that's where we're going to go. And again, we have sort of various pieces of these in various stages of progress. And to start, I'm going to tell you about this kind of mechanical perturbation experiments with some modeling to kind of get started. So this is kind of our first real results section. And this is the work I mentioned that's the collaboration with Mary Elting and Sharonda LeBlanc. And this is in a paper from earlier this year. And we started using cerulinin, so this drug treatment that restricts area expansion by inhibiting fatty acid synthesis. And Mary's lab was able to reproduce these kinds of buckling experiments that had previously been seen in the published work that I showed you. So time here is in minutes and seconds. These are showing a GFP label on tubulin, and they're contrast inverted, so where it's dark, that's where the signal is. And Mary did some kind of just additional quantification of this. So for example, here's a comparison of, as a function of the length of the spindle, what's its curvature, okay? And you can see that in control cells, so no drug, as the spindle elongates, there's maybe a little bit of curvature you can see in the beginning when it's assembling, but the curvature stays quite low. Whereas the gold with the cerulinin treatment, starting at a length of about 5 microns, not every spindle curves, right? You can see there's a big distribution, but on average, there's significant curvature of the spindle. And associated with that, if you quantify just how is the spindle elongating, okay, the cerulinin treatment slows it down in anaphase. So this is spindle length versus time, and t equals 0 is marking the time of the anaphase B onset, where the spindle that's assembled starts elongating more rapidly. And the blue curve here is control, the gold is with cerulinin, and you can see it's a significant, on average, slowdown, right? So this really suggests that the increased force of this envelope that can't expand in area is having an effect on the spindle elongation, right? So this is really consistent with increased tension of the envelope when it can't expand. And we'd like to understand that a little bit more. Okay, and so to do that, we're going to start with kind of a simple model to kind of estimate what this tension might be. And to do this, like I said, we're going to start very simple, building on some work that Francois Nedelec did about 10 years ago from this Ward et al. E-Life paper, where he actually built a model of multiple microtubules and motors mimicking the pombe spindle, and then, like in the upper right here, put a spring to pull on the ends and see what does it take to buckle the spindle, okay? And he found that, as you would expect for Euler buckling, that this buckling force that's required is proportional to 1 over the length of the spindle to the 4th. And using, again, just parameters that he found were a good fit for wild type pombe spindles and applying them here, for the 5 micron length where Mary consistently sees buckling starting, that force is about 31 piconewtons, okay? So we have an estimate of what that buckling force might be. And what does that mean then for the nuclear envelope, okay? And if you think about force required to deform a kind of vesicle-like Helfrich membrane like this, there's a characteristic, of course it will depend on exactly what the shape is, but some work from Dureni et al. In 2001 with Jacques Pro and Frank Ulicker showed in thinking about models of pulling out a membrane tube that there's a characteristic force square scale that's proportional to the square root of sigma kappa. And this notation here that I'm going to use, sigma is the surface tension, kappa is the bending modulus, okay? And kind of that form you could just get by dimensional analysis, right? That's the way you can use those parameters to get something with units of force. And then the prefactors, again, have to do with kind of the minimum energy deformation that you would get if you start pulling a tube. But still, we can use that to just do some estimates. If we use the numbers that we had before for kappa, from the Greg Huber work, this would suggest that the cerulinin treatment with this force is increasing the surface tension by about a factor of 20. So a big increase to about 0.3 piconewton per nanometer. Okay, so good. again, that gets us in the direction of starting to do something quantitative. How could we go further with this? What additional tests could we do? Well, to do this, we are going to start looking at spindle ablation experiments that Mary did. And this is another interesting thing that happened in this conference, where several other talks mentioned things about ablation. But it turned out that, so I started getting kind of excited, but it turned out that that's a technical term in machine learning training, right? Where you take out part of your model, I think, if I understand from searching it correctly, and then see what changes. So anyway, that's kind of interesting. It's kind of fun being outside this field and having this perspective that like, okay, well, we can't say we just simplify our model or drop a term. We have to call it ablation. Like, okay, let's make terms that are really dramatic. I think maybe machine learning likes dramatic terminology. Okay, but just to be clear, that's not what we're doing, okay? Here, when we're talking about ablation, we are destroying things with a laser, okay? So this is like Death Star ablation, okay? So Mary does these great experiments where the little lightning bolt is, okay? She's using a focused pulse laser to break part of the spindle. And now notice that the time here is in seconds, okay? And 0 is the time of ablation. So you'll see there's a little gap in the spindle right here. That's partially from the ablation and partially from bleaching. But after that, there's broken half spindles that then move together, and in some cases it can reassemble, which is kind of cool, but we're not going to talk about that. And so you could say, what happens if you do the same experiment in cells treated with cerulenin? And that's down here in the lower right. Notice this bowed spindle, right? So cerulenin has been added, the spindle is buckling, and she can ablate it right there. And notice that the two half spindles straighten out, and then again, they move together. And they move together more. So this quantification is showing after ablation, what's the change in distance between the pole separation. So initially they were a certain distance apart, how much do they move in? And with cerulenin treatment, that's bigger. Again, that's consistent with higher tension in these cells. Okay, good. Well, how else could we look at that? What could we do? Well, we're going to do, or really Royal We, Mary's lab, is going to do the same type of experiment with laser ablation, but on the nuclear envelope now, okay? So in these experiments, let me orient you to the markers. GFP is being used for 2 labels in these cells, okay? It's on a GFP NLS. So that means just the fluorophore with a tag that's a nuclear localization signal that tells the cell, bring this molecule into the nucleus. So when you see these images and it looks light outside the nucleus and dark in here, that's the GFP NLS, okay? The spindle is also labeled with GFP tubulin. And in these first images, you can't really see that. But if you look later, you can see our usual line that's showing the spindle. Okay, so both of those markers are there. And what happens after ablation, you'll see, maybe in the first second, there's not a lot. But then over a time scale of about 10 seconds, you'll notice that this GFP NLS signal is getting fainter, okay? While the spindle, this might be a little bit hard to see, but if you look, for example, in this 2.8 second image, you can faintly see the outline of the spindle and you can see that it's bowed. You can see that from the shape of the nucleus as well. The spindle is straightening out. So what's happening is ablation has made a hole in the nuclear envelope, and this GFP NLS can now leak out. So the fluorescence is decreasing. And as that is happening, the spindle is also straightening. And again, this is consistent with the idea that tension from the nuclear envelope that causes the buckling can be relaxed once it's been blasted open, right? Okay, so we want to look a little bit more about that and understand it. Before we do quantification, I just want to show you, here's some imaging showing what happens after ablation when you look at a nuclear envelope marker, okay? So this is with cut 11 GFP, like I showed you before, that stains the nuclear envelope. And this is a little bit subtle, but if you compare right at the time of ablation to about 30 seconds after, okay, and these are superimposed in the cyan and magenta over here, you can't see exactly where the hole is because ablation also causes extensive bleaching in that area, okay? But what you can see is that the shape starts to relax with the nuclear width decreasing, consistent with tension, some mechanical change that's decreasing the tension on the envelope. All right, so in doing some quantification on this, Mary's lab observed something really interesting. these two curves that look like they're right on top of each other. And these two curves are quantifying in gold the curvature of the spindle, okay? So this is one of those buckled spindles with cerulenin, and after ablation, the curvature goes down. And then in the pink is being quantified the integrated intensity of the GFP NLS in the nucleus. And so again, it's high at first because the nucleus was all filled with that marker, and then it goes down because it leaks out. And there's two things that I want you to notice about this. So first is that, so t equals 0 here is always when ablation happens. At the beginning, not much happens. There's a little bit of a lag, for maybe 20 seconds or something, right? And then these two things decrease similarly. It looks exponential-ish, and the time scale is very similar. This is just from one experiment, but if you do some averaging, and here, this is an average of about 20 cells, and we have kind of the units here, curvature, units are shown on the left, intensity, which are arbitrary on the right. These curves show a very consistent exponential decay with the same time scale. And I'm going to try to argue that I think it's a little bit confusing why that would be. It's just not clear why these should relax on the same time scales. And then I also want to notice that if you just measure how long is this lag, right? So from the ablation time where you start really seeing a decrease, the lag time for the start of nuclear leakage and for spindle relaxation, they vary somewhat, right? But they're very highly correlated. These two things really seem to be changing together. And we wanted to understand why that was. And I think it's confusing for reasons that I will explain now, okay? Because when I first looked at these data, I thought that this would be a great problem in which to figure out how big is the hole that was made in the nuclear envelope from looking at how the intensity decays, okay? And what I had in mind was this kind of great paper from Grigoriev et al. From 2002, where they solved exactly what are the kinetics of diffusion of a particle that escapes through a small hole. Okay, so imagine we have our nucleus. It has a volume V, and there's a particle like our GFP and LS diffusing around in there. And then there's a little hole of radius r that it can escape through, right? Okay, well, guess what? This happens exponentially, right? The number or the total intensity that would be inside will decay exponentially. And the time constant for that you can calculate exactly. It's 4 times the diffusion coefficient times r over v, okay? So I think we can measure everything we need to do to figure out this hole size, right? So 1 over k is that time scale of the exponential decay. It's about 10 seconds, great. The volume is about 14 microns cubed. We just say, okay, we're gonna say our unperturbed nucleus before we started this was a sphere of radius about 1.5 microns. And the diffusion coefficient, Sharonda LeBlanc can measure because she's an expert in fluorescence correlation spectroscopy, which I won't talk about all the details of, but it is a way that you can quantify diffusion. And here on the right is an example of one of her curves, okay? When you average across multiple experiments, that turns to be about 11 microns squared per second. Okay, so this is all great. We know everything. We're gonna get the hole size. What do you think it's gonna be? Anybody wanna predict? Order of magnitude? How big is the hole? I'll give you a hint. It was blasted with a laser. Alex is gonna volunteer. 200 nanometers, right? It should be kind of a diffraction limited spot type length scale, all right? So let's calculate it. These numbers say it's 30 nanometers, okay? This does not make sense, right? Again, given that it's an optical experiment, I do not believe that the hole could possibly be that small. It's not physical. It doesn't make sense. Okay, so we're definitely wrong. This calculation is not what we want. Any guesses as to what we're doing wrong? This is like one of those murder mysteries. You had the clues all along, but did you know what it meant? Any guesses? Okay, I'll tell you. This protein is being imported into the nucleus, okay? We did not think about the effects of nuclear transport, okay? The fact that this GFP has an NLS on it means that the cell is constantly pumping it in through nuclear pores, okay? And there can also be some passive transport out through nuclear pores. that at first we thought this wouldn't matter, but I'm going to try to argue that actually you really need this to understand what's happening in this problem. And then once you do, things make much more sense. Okay? So let's think about fixing up our model to take this into account a little bit better. We could think about what's the concentration of our GFP NLS inside the nucleus. So N here is going to refer to nucleus. And it can exit by two mechanisms. It can escape through the hole by the mechanism I talked about before. It can also have some passive export through nuclear pore complexes. And then the cytoplasmic pool can be pumped in by active nuclear transport, right? And then the balance of these could change over time as this hole size potentially changes. Okay, so you can do, so first of all, there are a couple really nice things you can get from thinking about this model. The first is that just having this first term with two sources of export can explain this lag that we saw in the data that I was really struggling to understand. Because if there's just a base rate of this protein exiting the nucleus, in order to see leakage, if the hole starts really, really small, it would need to get big enough that these two terms are comparable, okay? So that could help tell us something about the leakage. And then in addition, if we think about before ablation, okay, where this term would not be there, right? We wouldn't have a hole. We would just have active import and passive export. That actually can be used to constrain the ratio of intensity between the nucleus and the cytoplasm, right? Like how well are you pumping this protein in versus how it passively leaks out. And that we can quantify really well. Mary measured it, it's about 23, okay? That tells us the ratio of these rates. So this is gonna be great. We're gonna have almost everything that we need. Based on the nuclear transport literature, the time scale for this active import is in the kind of 10 millisecond range, probably 10 to 30 millisecond, where we're thinking about the time for a protein that's outside to diffuse to the nearest nuclear pore and then be imported. And so then this ratio tells us that the time scale for the passive export is in the range of a fraction of a second, okay? So this is starting to really make sense because remember our lag time? It was right in this range, right? It could be sort of maybe up to like a few seconds, but more of like a realistic time scale. And then if we do that calculation and say, well, how big does the hole have to be for us to really start to see leakage? We get a number for the size that actually makes sense. So something in the range of 0.5 to 1 micron is where we're really going to start to see a significant intensity decrease due to diffusion out of this hole. Okay, so what is this telling us? So what we think is that exponential decay is a result of the hole in the nuclear envelope growing over timescales of sort of tens of seconds, right? So there's an initial lag as the ablated hole starts to increase in size. And that is because the hole must be large enough for significant leakage to happen versus just what would happen with nuclear transport. And that hole growing significantly is also what's allowing the tension to decrease enough for the spindle to start to mechanically relax, okay? So I think what we're really seeing is dynamics of the increase of the size of the hole in the nuclear envelope. So this time scale, going back to these curves, we interpret as hole growth. And that actually is interesting and was a little bit surprising to me. What this is saying is that nuclear envelope hole sizes can change relatively slowly, right? The time scale here is kind of minute, like over tens of seconds, it can be slowly varying, right? It's not necessarily super rapid. I think we've learned something there. Okay, so what else can we do? So this is work that we published. I want to give, again, partially for Scott and Radhika, one example of some new work, just recent preliminary data from the last few weeks. We wanted to start thinking about what happens when you combine perturbations of tension, but also other aspects of the spindle. And one thing we looked at was ACE1, which is part of the PRC1 MAP65 family of cross-linking proteins in the spindle. So this helps cross-link microtubules. When it's not there, the midzone where antiparallel microtubules overlap gets destabilized. Spindle's elongation is slower, it's less stable, they fluctuate more. And so we thought this would be a great perturbation because if the spindle's already kind of not doing that great, not elongating fast, more unstable, having high tension from the nuclear envelope due to cerulenin treatment should really mess things up. It should go kind of bananas, right? That's what we're expecting. What we saw is in fact the opposite and was quite surprising. So in these curves, there's a bunch going on here, but let me show you. Focus first on the gold and the blue, which are gold is cerulenin treatment, and the light blue is ACE1 deletion. So they both show a lower slope, so slower spindle elongation by themselves, okay? But when you put them together, that's the pink curve, there's actually a partial rescue where spindle elongation is faster and looks closer to wild type than either of them individually, okay? So this was very surprising. I did not expect this. But to understand it a little bit better, Mary did some initial analysis of the curvature. And what you see, if you remember this plot from before, this is curvature versus spindle length. The gold is the cerulenin, where you see the significant buckling. When you also have ACE1 deleted, the spindles are not buckling like they were before, okay? I would have expected that to be worse because, again, the cross-linking normally would give the spindle mechanical integrity. I would think it's all kinds of messed up. But no, somehow it's actually getting better. The spindle is getting stiffer. And one clue to what might be happening here, you can see in some data quantifying the overall intensity of tubulin in the spindle. In the combined perturbation of cerulenin and ACE1 deletion, it gets higher. So something is happening that when the spindle is missing this crosslinker that normally stabilizes it, and it has this increased tension that we think puts more force on it, it's somehow able to get more microtubules and do something that is mechanically reinforcing it. And we do not understand why, but I think it's kind of an interesting thing that we'd like to get to.
Yeah.
Okay, so this is all kind of warm-up estimate type modeling. I want to show you a little bit of our data on building kind of a better model of spindle-nuclear envelope coupling. And this is very much work in progress, so I'll show you some initial results. So Will Lowe, who's a postdoc in the lab, has been implementing some approaches used in other work for a dynamically triangulated membrane model. Okay, and let me kind of show you the video of just kind of something starting from a sphere and kind of evolving with not much happening. Okay, so again, this is a Helfrich type model. There's bending energy and surface tension, approximate volume conservation. The membrane is fluid and undergoes local thermal fluctuations with local drag. And there's some special things you need to do to kind of maintain mesh conditioning in these models. Okay, so this we can kind of integrate with our other Brownian dynamics type modeling to couple to the spindle. And here's kind of just an initial example simulation that Will did that's showing how this kind of membrane type model can distort. So here this is changing not because of the spindle, but just because of force that we put into the simulation that separates the two ends. But you can see that it can deform, can separate, it can pinch down, and you can also, I think, the thermal fluctuations are visible here. So starting with this, Will is building an anaphase spindle and nucleus model. So incorporating this membrane model along with a semi-rigid rod that represents the spindle, and then kind of complexes that represent the spindle pull bodies and the chromosomes that help distribute the force along the surface of the nuclear envelope, because we know that that's important. And in our spindle model, to start at the moment, we're thinking of this as a rod made up of two half spindles with an overall length and an overlap length small, capital L is the overall length, and an overlap region of length small l, where the spindle changes length because of sliding forces proportional to the length of the overlap that extend it, and then can shorten due to compression from the poles. Whereas the overlap changes length due to sliding if the spindle as a whole changes, but also due to polymerization dynamics, kind of dynamic instability for the microtubule bundles in the overlap. Right? And so again, this is very preliminary. We just got this working relatively recently, but I want to show you one example simulation. Okay, so here the blue is showing the nuclear envelope. The kind of gold blobs are showing the SPB chromatin complexes, which for now are rigid. The spindle is elongating under this sliding and dynamic instability model. You can actually see the two halves that are orangish and greenish, and then the overlap in the middle. And you can see, again, although this is kind of very preliminary with rough parameter estimates, a series of shape changes that at least qualitatively are quite similar to what we see in our yeast. Okay, so we're really excited about this and kind of currently working to make more like quantitative comparison and then start to use it for prediction. Yeah, Alex, please.
Why do you want to bother with thermal fluctuations? They're probably inconsequential here.
Yeah, so how important thermal fluctuations are, I think, is not that clear. So we had already kind of implemented Brownian dynamics type simulations for other spindle components where we know, for example, motor diffusion and binding kinetics are quite important. And we want to go toward having everything in the same framework.
Because
So
you can.
yeah, and because we can, yeah. But we'll see. Actually, how important they are is something that we have a little debate going about, but we'll find out. So let me just show you an example of playing a little bit with parameter variation in the model. So this is kind of a grid of simulations where as you move to the right, the size of this SPB chromatin complex gets bigger. And as you move down, the sliding force that you have from motors gets bigger. And you can see that as you vary these parameters, the nature of the distortion and how symmetric it is changes. All right, so let me go back and just show that again so people can see what it's like. In some cases, like in the middle here, you can see that the elongation initially looks fairly symmetric, but there can be transitions where then most of the nuclear volume winds up on one side and the other one is smaller. So for example, okay, good. So again, this we're just in the process of developing. Can I check on time?
Very close to the end.
Very close to the end. Okay. So I am just going to really briefly talk about this as kind of a teaser for future work and to leave you with some interesting puzzles. So we recently have been developing something called spindle protrusions as a kind of novel window on force in the spindle. So the idea is that with the right genetic trick, you can get spindle microtubule minus end anchoring to be perturbed. And microtubules or microtubule bundles can slide kind of past the end of the spindle, and that can distort the nuclear envelope. Okay, so we've done a bunch of work on this, which I am not going to tell you everything about because there's not. I just want to show you a couple. There's modeling, blah, blah, blah. It's great. Just a couple interesting things. So we have been thinking about this. So here's some imaging. The green is showing the nuclear envelope in spindle pole bodies. Red is showing the spindle. And here's an example of kind of something we've seen and we've studied and we think we more or less understand, that sometimes you can get little membrane tube-like protrusions that deform the nuclear envelope. So there's one there. Here's another one over there. I hope you can see them, especially in the green channel, right? They look like these little lines. Okay, so this is great. This is what we expected. This is what people had seen previously. And we think that we understand something about their mechanics with some modeling I'm not going to show you. But then as we looked at these some more, we can also sometimes see some things that are kind of weird and not what we expected. So here in this cell, there's a short spindle, and it has two protrusions, one on the top and one on the bottom. And what you start to see over time is that these protrusions can start to rotate relative to the spindle axis, and they can make nuclear envelope shapes that are not what I expected. So like notice how on this side of the protrusion, this is pretty straight, but then on the other side, there's kind of a big rounded part. It doesn't look very symmetric. And I don't really understand that. I would like to. And this can also drive, in addition to the shape asymmetry, rotation of the spindle, right? So in the beginning, you might notice the spindle here is relatively vertical. Later on, it's tilted to the side. Okay, so the interactions of these protrusions with the nuclear envelope can exert mechanical force on the spindle that can rotate it and also strain things with the nuclear envelope. And I'll leave you with one more example that I think was even more surprising, at least to me. So again, here is a spindle, and it has two protrusions, one above and one below. Both of these protrusions, upper and lower ones, start to rotate. And it can lead to some really puzzling and asymmetric nuclear envelope distortions. So I'm highlighting this one, which looks like Antarctica, right? So there's kind of a protrusion going up to the right, and then for some reason there's this little dip in the nuclear envelope before it extends after that. Or this other one over here that looks kind of like a potato with a shoot coming off of it. Again, because of the protrusion, it makes sense that there's this distortion up here, but how is it affecting the shape on the other side of the nucleus? That's something that I don't currently understand, but we would like to. Again, getting more kind of dramatic mechanical deformations of the nucleus using this as a tool. Okay, so I will just leave out these conclusions, what I've tried to just show you is evidence of significant spindle and nuclear envelope mechanical interactions, development of some new tools to characterize how they interact, starting to kind of quantitatively do some estimates of what this force and nuclear envelope tension and pore size dynamics can be. And we want to work toward more integrative modeling of this. And I'll thank our funders, and I'd be happy to take questions. Yeah, please.
I have like two questions. One is essentially when you remove this AAC1, you showed that the microtubule number sort of increased, or the experiment hints towards that.
Yeah.
So I'm just wondering, is AAC1 some sort of a polymerization suppressor? So when you remove it, like microtubule goes up, or what happens?
Yeah, okay, so the question is about, would you expect removing ACE1 to lead to more microtubules in the spindle? Does it depolymerize microtubules? And no, in fact, the evidence for ACE1 is the opposite, that having their help stabilize microtubules and increase their number. So why you end up with more microtubules when it's deleted, and then also you have cerulenin, we don't know. Yeah, we just don't.
And like, I have a second
Sure.
question. So in your like nuclear envelope deformation model, like do you consider target pressure or like fluid exchange between the cytoplasm and nucleus, or is just the membrane tension?
Yeah. Okay. So so the question was about volume and turgor pressure in our nuclear envelope model. So what we see experimentally and then also in our simulations is that the interior volume of the nucleus stays approximately constant, but the area has to change pretty significantly given the weird non-spherical shapes, right? And so given that, we have started with just keeping basically a volume conservation type term and then letting the area vary with the surface tension. And so far that seems to do okay. But we'll see. I think that's another interesting question that we'd like to understand as we do more comparison with the data.